Okay, so welcome everyone, and today I'm very happy to that we have Jill here, who will present her work. So you said it was done at Utrecht first, but now Jill is working in Charité in Berlin, and it will be a nice talk about how to use network science for neuroscience. Okay, so Jill, the virtual floor is yours.
Thanks, Carl, for the introduction. Hi, everybody. My name is Jill. About a year ago, I started my second postdoc here in Berlin. And before that, I spent 2.5 years in the Netherlands in Utrecht at the hospital, working at the ALS Center. And the work I present today is mostly from Utrecht, where I spent a lot of time talking to neurologists and neuroscientists and so on about network science. So I'm really happy and feel honored to present it to you today. Maybe just shortly about myself. My background is
never mind.
Hi, I'm a mathematician. I studied mathematics in Munich at the Technical University. It was quite pure mathematics. And then in my master's, I discovered financial networks. So I thought, oh, networks are cool, but the finance part I didn't like. So 8 years ago, I decided to go into network science as a mathematician. As Carl knows, a very brave move. And I'm surviving still. So let's see. I will give to you a talk about mostly the paper of Connectome-based propagation model about ALS. I will very slowly slide into the topic. So if anything is unclear, I know there are different jargons used, so just feel free to ask. And then in the end, I will give a short outlook on my current work here in Berlin, where we are using a virtual brain to stimulate it and see if we can change the dynamic regimes for patients. So I hope in the end of the talk, you can answer how a random walker can help patients. So first of all, let's get this straight. In case not everybody, I heard there are various backgrounds. Not everybody maybe knows if I say brain networks, what does it mean? What do I mean by that? Mostly today, it will be about the structural brain networks. So that's rather easy to understand. You have a network where the nodes are brain regions. Like here, for example, the motor cortex regions is here located. It's a center for all your movement where it's directed. So nodes are regions, and the links in between two brain regions are actual anatomical connections between those brain regions. So those, like here, those are called fiber tracts or white matter pathways. These are long-range connections, in this case, connecting the motor cortex of your left to your right hemisphere. And how are they found, those connections? They're actually found by parcellating the brain in different regions and then looking at those fibers that always look very nicely, where we just track water molecules and then assume when a water molecule can move along this way in an MRI scanner, we assume that there is a street or that there's a pathway. And then also we here, as you can see, we distinguish different regions in the brain. This is called a parcellation, and the regions become nodes, and the links then become the links of the structural brain network. So a structural brain network, again, the nodes are always regions here, and the links are just the streets in between the regions where activity can be propagated, and firing can go from one to another region. So just to show you again the overview, we often start with MRI, so I will talk about MRIs mostly. The MRI pictures look like this on the left side. Sometimes they look very blurry, and you normally, like if you look at it yourself, you wouldn't really trust this data, but there's a lot of pre-processing steps where they cancel out the noise and where they really clean up this data, such that we in the end have an individual structural brain network. So this is called the method of finding those fibers, finding those links is called diffusion tensor imaging. And then often you get a T1 scan as a patient as well. This is a normal procedure. Whatever you have, whatever MRI of the brain you will get, you will have a T1 scan, which is a very high resolution scan, just looking at the structure, looking at the regions of the brain. And those regions, you can calculate the volume, for example. They call it cortical thickness, just the volume of this region. So you can get out a lot of different properties of your own brain once you got an MRI. And last but not least, there is a functional brain network. What is that? It's actually based on fMRI, so functional magnetic resonance imaging, where you would get sometimes just resting state, so eyes closed, not thinking about anything in particular, and still one would see that some regions light up like this in the scanner. These regions are then activated, and we record the signal of those regions as the bold signal. Bold is just here for blood oxygen level. So that's an indirect measure of activity of neurons. So if the oxygen in a region is high, it means that neurons have been firing there a lot. And then you would have, for each region, you would have a signal. And these signals you can correlate. So you can look at so normally they take two regions and they look at the correlation of those signals. If the two signals of those two regions are highly correlated, so they do the same thing, then we think those two regions are communicating. So they have a strong connection, not in the structural network, maybe there as well, right? But in the functional network then. So links in the functional networks are links telling us about how much those two regions communicate, how strongly.
Sorry, can I ask a question?
Sure, please.
It's just a quick one. If two regions have a strong functional connection, does that not imply at least that there is a strong structural connection, or can that be possible without it?
It can be possible without it. So that's what it makes it quite interesting. So you have the correlation that you have strong functional connections are often strong structural connections, but you also have functional connections when you don't have a structural one. And this is via indirect connections then. So just taking a longer path in the network and still being able to communicate very efficiently. This is what we sometimes have in the brain, for example, between right and left hemisphere. There's rather a long way, but those regions still in right and left hemisphere fire quite simultaneously. So you will see that it's nearly happening same thing, right and left. And yeah, so you have strong functional connection, but a weaker structural one.
Thank you.
Yeah, you're welcome. Yeah, so today we will focus on the structural part, which is easier, I think, to grasp, but later on I will tell you about the functional part, which is as much important as this. And my PhD thesis, Carol knows, this was actually about the relation between structural and functional networks in the brain. So there are many publications about that. It's still an open topic of discussion. Okay, so let me tell you and explain to you what ALS is. It's short for Amyotrophic Lateral Sclerosis. And when I was working in the Netherlands, people would ask me, what is ALS? Everybody would be like, I've heard of this. And then I would just mention the name Stephen Hawking. People would be, okay. And they would have already quite a good picture of what is happening in the body if you're getting ALS. So it's a horrible disease. It's not stoppable, no treatment, no drug at the moment, only one approved that's prolonging your life for 3 months. That's not a lot. If you look at the median survival, which is 3 years after disease onset. So getting the disease and the diagnosis of ALS gives 50% of the patients a chance dying before 3 years and another 50% of surviving those 3 years and living up to over 20 years, or yeah, you know the story of Stephen Hawking, much longer even. So it's very heterogeneous. You have such a different picture across the whole spectrum. And it's still very well known, even though it's rare, as you know, probably the Ice Bucket Challenge. This is my former boss, the Professor Leonard van den Berg in Utrecht. He's the head of the European ALS Center. And of course, participated as well a couple of years ago. You must remember they raised a lot of money, and also my postdoc was partly funded by that, so which is pretty cool, I think. And you don't have to go all the way to Utrecht to find experts, but we are closely collaborating with people in Oxford as well. The lab of Martin Turner is the experts in neuroimaging and ALS, and they have also already looked at disease progression and trying to model it. So in ALS, your muscles will just become weaker and weaker, and the healthy motor neurons get deteriorated, get damaged, and this damage is spreading from the motor cortex, which is the center for movement, towards other brain areas. Also, ALS patients, like 20% of them get FTD, so dementia symptoms. So it's not only that you lose your muscle power, but it's even more horrible. The cognitive function can also suffer a lot in ALS. So yeah, a very bad disease. And in Europe, it's estimated that currently there are 15,000 cases only. So quite rare and not well understood. Yeah, gives us a nice gap to look into and use network science for it. So the motivation was that we got in Utrecht a large longitudinal data set of thousands of MRI scans already collected since 2009. Every patient, they just started scanning them every 6 months until their death, right? As long as they could lie in the scanner, they were scanned. So we know a lot, we have a lot of data on the progression of the disease, and nobody has really looked into that before I came. They have, for example, 366 ALS patients that have at least one scan. So if you look at the median survivor, you can think that you will lose patients also after the first scan. Sometimes a diagnosis is given much later because ALS as a diagnosis is in itself a challenge. You first have to exclude all the other reasons like MS or other diseases that could lead to the same symptoms. If you exclude them, that takes about a year. Only then you're getting diagnosed with ALS and included in the study. And then we have also 60 patients that had 4 scans. Between each scan, there's like 6 months. So this is a 2-year interval approximately. They also included healthy controls that were age and gender matched. So large data sets. And in ALS, the big question for each individual is, where in which group do I belong? Am I a long survivor? Will I survive very long? Or am I a fast progressor and will die within 6 months? So you need tools to tell the patients and forecast what's happening to them over the coming 2 years. So that's what we set out to do. We wanted to forecast the disease progression for each individual patient using MRI scans and network science. So the motivation behind was a bit looking at the literature. There were studies on post-mortem data where they, after the death of the patient, dissect the brain into slices and they look for a specific protein. So in ALS, it's p-TDP43, and they can tell how much this protein is aggregated in which area. And when you look at this, also from the network, you can see that those areas are strongly connected. And it seems that this protein, there's a hypothesis, is spreading in a prion-like manner. So along the white matter pathways as well. So we were like, okay, a protein spreading from a region to another region, what's that for us? That's a random walker, right? So first we need to make clear what is this network impairment that we are trying to model. We need to look at the data and we need to see what exactly is happening in the network of the patient over time. And then we will use a random walker to predict it. So for that, I need to shortly explain the method that we're using to just see what's the weakening of the links like in our network. It's called network-based statistics. It's was first used by Zaleski et al. In 2010, quite an old method, it's very well used. In neuroscience, I'm not sure if other network scientists know about it, but it's using a trick for multiple comparisons. So normally, if you look at the network, you want to compare 2 groups, controls and patients. Then if you want to look at the links, which we want to do, we want to look at those connections, and we really want to just see, is this connection weakened in patients already or not? So we compare patients' controls, and we want to find those links that are not strong enough anymore, not like healthy controls. So when we would do that for every link. We have 83 regions here, times 83 gives a huge matrix. So if you want to correct them for multiple comparison, you will not end up with any significant differences between controls and patients. Often the case, right, when you compare two groups and with their different weighted networks. But this NBS, or network-based statistics, is the trick which still will point you in the direction of the network, where a lot of those nearly significant connections can hide. So how is that done? You first do a normal t-test for every link. So always the weight of the link for controls against the weight of the link for patients. You test the distribution, students t-test against each other, and then you have a t-value after doing the t-test for all the links. So every link gets a t-value. So normally here you would just correct for multiple comparison, Bonferroni or something, you would find no significant differences, full stop. Yeah. But now we do a trick. We say we take a certain threshold, and we will actually take 0.01 here in the study. And we will look only at those links having a higher t-statistic than this threshold. So we actually take the p-value corresponding to the t-statistics, but it's the same thing. You think of a threshold, maybe it's more intuitive to think of the p-value and then a threshold. So look at all the p-values on the edges, and then you keep only those that have a lower p-value than 0.01. If you then look at what's left of the network, you can see that there are different components left. And we will always focus on the largest connected component, because we know the hypothesis in ALS that the deterioration of the network is spreading from the motor cortex along the links to other regions. So we are looking for something connected. We are looking for a connected component like this. And once we have this connected component, which is connected through all very low p-value links, then we count the number of nodes in it, so the size, and we compare whether this size, by just now randomly shuffling controls and patients into random groups, repeating the same thing, and then we look if this size of connected component was expected by chance or not. So it's just a trick to look for clusters of low p-values, let's say. And we did that. And this is how it looks for our data set for all the patients that have 4 scans. So the red links here are those links that came out of the NBS analysis. So those links having a lower p-value than 0.01 being in the largest cluster. So here's a cluster of low p-values. It's around the motor cortex, and it's over time growing, as we would have expected it, but now we have also the data to show it. So that's quite nice. It's here, the temporal lobe, so this can have to do with cognitive functioning as well. So it's spreading, it's not only motoric anymore. So this is based on the MRI scans and the structural brain network. This is nice. So we try to model this. This is a impairment that we are now trying to model with the random walker. So just to give you a very easy looking overview of what this whole study is about. We have seen this empirical impairment. We will call it empirical impairments, just what we see on the scans, which links are getting weaker over time. And then we will use a computational model and simulate impairment now. So we always said in the article computational model because it was written for a neurological audience. So I can tell you it's a random walker on a network. And then we'll try to compare this empirical impairment to simulated impairment. But this will not be all of it, as we will see in a second. But first, let's see how we do it with the random walker. We take the network for each patient from the first scan. We let the random walker walk over it, and we count how many times the random walker was in each region. So what kind of random walker is that here? So first of all, the random walker will only start in those Brecht Schneider or Bragg stage 1 regions that I showed earlier. So very specific regions where we know from post-mortem data the disease probably starts. So we'll put the random walker in each one uniformly random at one of those black regions. Then we let him take one step. And in the second step, it will already self-multiply. So from each region where the random walker was already once before, a new random walker will start as well. And it will spread, 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 and will reach more and more regions until the first random walker took 5 steps. So that was enough. We tried out different steps. And then all the random walkers will stop. We will count how many times for each region a random walker was in that region. So a region is here just a node in the network, right? And then we will have our number of times the random walker visited, or we will call it random walker aggregation levels, which more corresponds to the protein aggregation levels of this protein from the post-mortem data. Yeah, and when we do this, now we find a very high correlation between the random walker aggregation levels, so how often a random walker visits this region at the first scan, and corresponding to the empirical impairment at time point 4. So this is the main result, that we can take the first scan of a patient and we can tell him or her which of the regions will get critically impaired in 2 years. So 4 scans, 3 scans later. So how is this empirical impairment actually quantified? So here we took, for each link, we calculated a Z-score. So Z-score just comparing the weight of this link to the weight of the controls, because for each group we luckily have age and gender matched controls. So we can just say this is how the link should look like. And then we will calculate the Z-score. If the Z-score is negative, it means here that the link is weaker than in controls. So when we know this for all links, we want to look at regions, we will just count. So taking like the node strength of each node here. And we will just count how many negative Z-score links are attached to a certain region, meaning this region has a lot of links attached to it that are weaker than in control. So this is our empirical impairment, how you normally do it. So you see a very high correlation. You see that we can predict some impairment here, which is great. And when you have worked with random walkers before, I know many of you have probably, then you will say, okay, this is just a degree. It's just that the random walker will end up in all the hubs of the network, and this just shows that hubs of the network are impaired in ALS. Yes, that's partly correct. That result wasn't out there, so it's good that we clarified this, that regions which have really high connectivity are more likely or more vulnerable to the disease. But there's also more to that story, because when we correct this 4 degree here, I put that here to just, because it's great to talking about networks and to talking to other network scientists about this. When we corrected for degree, the relationship was weaker, but still there. So it has to do with the degree, but the degree doesn't seem to be the whole story.
Are these undirected networks or directed?
These are undirected networks. So the truth is that brain networks are directed. It's always directed. We can look at monkeys, we can look at mice with horrible track tracing experiments. We know that they're directed. So it's not necessarily the same from one neuron to the other as from this neuron back. But for MRI data, we always assume bidirectionality, because that's the only thing we can track. So we cannot zoom in that far to say which direction that connection is, unfortunately. So this is all bidirectional networks weighted. Yeah, I should have said that. Thanks for the question. Okay, so we have established that the random walker can predict something from an MRI scan, but that's not enough. And really not enough for the neuroscience community. So once we sent the paper, they were like, oh, great, you can predict something based on an MRI scan, you can predict something else on an MRI scan. But what you really need to do for us is predict clinical characteristics. You need to predict something that the patient can, or the doctor can see what is it and can directly relate to. So after here having the empirical impairment against the simulated, I showed you group analysis results. So this is just a group average matrix where I put the random walker in. But in a second, I will show you individual analysis as well. And then we'll go even further and compare our aggregation levels that we had. First was the pathological staging. So pathological just means this post-mortem staging. So we need to show that our random walker, what he predicts, those aggregation levels are actually in line with the aggregation levels I find post-mortem. This is often very important. And then we need to see if we can predict actual survival. If we can say, based on the random walker, how long a patient will survive. And if we can say, based on the random walker, which or how much of his limb strength, so these are neurological scores, that's just a score composing some clinical values. Can you still move your right arm? Can you still move your left arm, right leg, left leg, and so on? There are all those tests which end up being a score, and I will show you the results that this also corresponds to those neurological scores. So it's not something very abstract, but we can pinpoint directly, in more detail what will happen to the patient. Also, the validation with an external data set we will do. That's often very important because you can have the best data set of ALS data in the world, which this currently is. But if it's not working for others, it's still not a good model. So we first let's cover the correspondence with the pathological, the post-mortem staging. So here you can see again the Brettschneider stages, and you can see our aggregation levels. And if we look at all those regions of the stages and we look how often was the random walker there to visit them, we can see this trend going from stage 1 to stage 4, meaning that if the random walker was there more often, this region, it corresponds to a region which also post-mortem was found to have the most damage. So a visit by the random walker in this case is bad, just as we showed before with the empirical one. So it's good that it corresponds somehow. So we tested the trend here, and the trend is in there, so that's great. It corresponds somehow to the pathological post-mortem staging. However, post-mortem, everybody is just in one stage, right? So they cannot really do in vivo staging so far. So it doesn't have to be a one-to-one correspondence. It's completely different, right? Once you're dead, you've either gone through a couple of stages or not. So yeah, it's difficult, the post-mortem. So it's better to have in vivo, then you can tell a person which stage of ALS they're in when they're still living. And then we took the individual level, as I said. So before that was a group level. Now this is the random walker aggregation level just averaged over all the individuals. Looks quite similar than to the group level. This is the empirical impairment averaged over all individuals. And this is a Z-score again. So low Z-score means very weak connection compared to healthy controls. So you see already the correspondence. Here you can see it just for one individual patient. Of course, yeah, I took a very nice patient having a high correlation between his simulated impairment and his empirical impairment. Same thing as before, high correlation. And then when you look at a histogram of all the different correlations of all the different individuals, you see that we have a high mean correlation. So on average, we reach a high correlation also for these 208 ALS patients. So that's always important, that you don't only have something that can predict something on a group level, but you need to go to the individual level. So in ALS, it's especially important because ALS patients can have such different disease progressions, such different disease courses that, yeah, I think we need to find something for each individual. And the external validation was done with just 30 controls and 30 ALS patients. So that already shows you We have quite a great data set. So here's from Dublin, and they could offer us 30 patients that had 3 scans, which were done with the same protocol. And we were able to show same things here. You see already similarity of those plots with the other ones before. So that's great. It worked on an external data set. That was, let me tell you, that was very exciting to try out. And I encourage everybody to do so and include it in their studies, but often it's not done for obvious reasons. And now can we predict those clinical characteristics? Because seeing something on an MRI scan, great. As I said, you can predict it, something from an MRI scan to another MRI scan, but what does it mean for the person? So first of all, survival. My PhD student, Hannelore van den Burg, she did an earlier paper on machine learning and just predicting survival based on some clinical characteristics like the age of the patient, whether he has dementia symptoms or not, and just putting in the whole structural connectivity matrix and the volumes of each region and predicting whether a patient will survive shortly, will be in the medium survival class or the long survival class. So what is meant here was, so it's a 3-class prediction, it's a deep neural network. Short just means the patient will survive under 25 months after symptom onset. So anything onset, weakening, twitching in his arm, muscle weakening in his hand. Medium is between 25 months and 50 months after symptom onset. And long survivor is everybody above 50 months. So you give in the clinical characteristics of an individual and his structural connectivity matrix and another volume measure, which is quite important for ALS, and you can predict this successfully. This was done on 135 patients. And now, a few years later, we had much more data because we were following these patients, and only when patients are either living longer than 50 months or having died, right, then you can put them in a survival class. So we put in total 366 ALS patients in the survival class. And we did that with the previous model, plus now the random walker aggregation levels. So we added them as an additional input to the deep neural network. And again, we did the same thing. We tried to predict the survival class, and we looked whether the random walker can help us to predict survival. So here you can see that we divided these 366 ALS patients in training, validation, and test data set. And the results for the test data set, if we just take the random walker, nothing else of the others, we get an accuracy here of 64.8%. So 64% of patients were classified in the right category, short, medium, long survivors, when we just give as an input the random walker. So that was giving us already like a good feeling. This is, so chance here would be 33%, right? So we have 3 classes. So anything above that is already good. We were very happy with 64%. And then we combined this network. So going back again, we took everything here before just a 1, 2, 3, and then we added the random walker. And if we put just the 1, 2, 3, we got an accuracy of 79. If we add the random walker, we get 83.3. So it increased slightly, but significantly when we add the random walker to this machine learning algorithm, which predicts survival of ALS patients. So there seems to be something that the machine learning is not getting out already of the structural matrix just by itself. So that was great, but then this is also network science, a random walker, and then on top of it, machine learning. And we were submitting to a neurological journal to reach the audience of neuroscientists, right? So we needed to do more. And we did also look at those neurological scores. So here on the upper right corner, you can see how that is performed. Maybe you had that once, who knows, testing reflexes with a small hammer. And then when you're an ALS patient, you will have often hyperreflexia, that too much of a reflex will show that actually it's not, something is not correct with this limb or with this part of your brain then as well, or spinal cord can be. So you get a score. And I mark this here as bad. So once you get a score, we call it this neurological examination score. This called upper motor neuron score, or here UMN, just to abbreviate it. So let's call it neurological score. So if your neurological score is high, it's bad for you, meaning that you have a lot of things not working. It's done to your arms and to your legs and many other parts where they were tested. So when you have a high score, we found a relation here that also looking at your motor cortex, so that's a region for movement, right? Looking at the aggregation levels of the motor cortex, so how often did the random walker visit the motor cortex, corresponded to this bad score. So a Z score of the aggregation levels of the motor cortex just means that the random walker visited the motor cortex in these patients much less than the healthy controls. So if the motor cortex is not visited that often by the random walker, if you think about it, you get a bad score. If you think about it, it makes sense, because it would mean something that the connection to the motor cortex is weakened, because a random walker in our case chooses based on the link weights where he has to go. So if he doesn't go there, it's probably because the connections to the motor cortex are already weakened. That could just be an explanation here. So we showed that the random walker can predict the clinical score that you have 6 months later. So this is based on the first scan. This is the second scan. It's because not for all the 4 scans, we had this neurological examination, but only for 141 patients, which is still quite large if you know other studies from neuroscience. And another thing we found, which I found quite interesting, is that it does not only depend on whether the random walker visits the motor cortex or not. You can also just look at the standard deviation of the aggregation levels of the random walker. So the standard deviation just means that your brain can look either like C or like D. So what do I mean by that? So patient here in C, patient C, let's call him patient C. Patient C has a high standard deviation, has a lot of variation. The random walker comes to some regions much more often than to others. This is a healthy pattern. As we can see here, he will get high standard deviation. He will be up here. That's actually this patient here. C is this patient here. Could have marked that, but he will have a very low neurological score. Low is good, high is bad, right? It's a bit counterintuitive. But the patient's brain will look like this after the random walker visited it. And this will give us an indication that 6 months later, he will be still quite fine. In comparison to patient D looks a bit like this. So the random walker visited many more other regions as well, not only the motor cortex, went everywhere in the brain. So if you think this is aggregation, this is like the disease spreading, this is bad, right? And you can see this from the total scores as well, that this patient having a low standard deviation, all the regions having similar aggregation levels, will end up with a bad score on the next appointment. So this if you see this the first time, it can be quite confusing. But just to sum up this slide, we have a relationship with different clinical characteristics with the random walker aggregation levels. So we were very happy that we found this, and by this we were then able to get this into the review process. And just a summary now of the things that I told you about this study, then I will have shortly time to give an outlook. We compared with the postmortem staging that worked. We showed that the random walker helped to predict survival. We compared it with neurological scores and found some relationships. We showed this for group and individual analysis, and also we validated with an external data set. Just as a conclusion and a future pathway here of this research is that we showed that the random walker has potential to forecast the disease progression. So computational network models can really help to look into the future of a neurological disease. This another model, but also diffusion model, has been used for dementia, and there it worked quite well to predict cognitive scores. So I think there are many other diseases out there that we could model like this, and yeah, I think it's worth doing it. And also in clinical trials, we should use additional biomarkers. So far, they often use like questionnaire-based data and survival to test for ALS whether a new drug works or not. And this can give you like a window in the future already, and should not be used as the only biomarker. I don't say that, but as an additional biomarker can give you an indication for what kind of a patient are you dealing with. And similarly, I always thought like, okay, we have done a forecast of structural network, but we want to also look into function and with dynamics, right? And then we want to look at cognitive scores. So there are many more aspects of this disease out there and of other diseases as well. So I felt after this postdoc a bit like, okay, I should do something with dynamics. Let me just first thank my colleagues from this study. We will have a discussion then also about that after I maybe just share a few of the newer work things. Especially Martijn van den Heuvel, who was my mentor for this whole project. He's now in Amsterdam. And as I said, I always felt a bit like this when I was looking and predicting structural damage in the brain. It's great that we can predict it, but what can we do about it? And my new goal is to use network science actually for therapy and use all the knowledge we have from the MRI and the dynamics of the brain to then change those dynamics and in the long run change also the structure of the brain to help the patients to stop this progression. So there I found the virtual brain, and that's the main topic that I'm working on right now. It's a website, thevirtualbrain.org. I encourage you all to download it. It exists already for over 10 years. It's just a web browser-based application, which you can just play around with, where you can just load a structural brain and you can look at the dynamics in videos already, and you can create a seizure in the brain, look at what's doing it to the brain. You can create different diseases to the brain. You can try and maybe try to stop them. And how does that work? This whole simulation platform is nothing else than a simulation platform, has the same structural network as the underlying basis. So nodes are regions and links are those anatomical streets. But then also for the dynamics in each node, in each region, we have a model, a neural mass model. So that's a model describing in more detail what's happening inside that region, what kind of firing is going on between the inhibitory and the excitatory population. So if you look at it from a mathematics perspective, it's just a handful of equations describing a dynamical system on each node. And then they are linked together via the structural network with some global coupling factor that you can just escape. It's just a scaling factor that you can make connections stronger or less strong. And then it starts firing and it starts simulating. And you can record then, for example, the bold signal, so an fMRI signal, you can record EEG, anything you can do virtually. You don't have to do to the patient anymore, but you can also fit the virtual brain to the patient's brain so that you make a copy of the patient's brain in a virtual environment and test out different strategies there. So I found that quite cool. That's why I came to Berlin. Let's skip over the equations here and look what they have done to it already. They are already using it for a trial with over 400 epilepsy patients, where those epilepsy patients are not only operated. So in epilepsy, when you're drug resistant, so no medicine helps, a region of your brain is surgically removed. It's taken out. So those patients are not only operated by the opinion of the neurosurgeon, but also by the virtual brain. So they're testing out, taking out those regions in the virtual brain first, and then they write a short report for the neurosurgeon, and he can consider that opinion of the virtual brain as well. So it's quite cool. So I joined this new project with Professor Petra Ritter, where we would try to just move the brain from the disease state of the dynamical regime perspective to the healthy state with deep brain stimulation. I don't think I have time for that. But shortly, deep brain stimulation is an intervention which works for many different diseases. But you have to program the stimulation device, which is implanted here in the brain, correctly to make it work for patients individually. So again, we have an individual prediction that is needed here. And this is just the interface of the virtual brain, I wanted to show you last, where we can actually then program such a stimulation. So it's nothing else in the structural network with the dynamics on top of it. And we can look at it here in just in the GUI of the virtual brain and extract all kinds of features from it and then compare it to the real data. Yeah, let me just thank my new colleagues. And any questions? Now.
Sorry, quick question on what you just showed. By stimulation, what do you mean? Is it just a release of hormones or?
No, it's an electrical device, really, the deep brain stimulation. It's an electrode looking like this with contacts, which is really giving electrical pulses to the region and can alter the dynamics by that. It alters firing patterns and then it alters the dynamics.
Right, thank you for a great talk.
Yeah, thanks. Let me just go back to my email. So in case you have any longer questions, you can always write me. But I'm free to discuss now. Yeah. Hi.
Ask a question. Hi, I'm Gesine from Oxford. A really exciting talk. So what I've learned about brains in another project is that there are these called brain states. We've been looking at people between awake and getting anesthesia, and there are different states. So these are sort of whole parts of the brain which are activated jointly.
Yeah.
Think of this as a random walk in varying environment, if you like.
Exactly. That's I think quite similar to this. What I mean here. Yeah. You have like the whole brain as a state. Yeah.
Yes, but so different. So like a hidden Markov model. So you might be able to do things analytically rather than just, well, I shouldn't say just, but rather than only simulations, because there are, as you know, drugs which affect different regions of the brain differently, and then you can choose which kinds of impairments they are happy they would negotiate to live with better than other impairments, for example.
Yeah, I think that's really exciting, and that's exactly what I love looking into as well. Whether we can skip all the simulation and do just some Markov modeling.
You probably want to do both, but for for explanatory or for strategic thinking, possibly some modeling might be good.
Yeah, I think you always have to combine there, especially when you want to convince the community.
Yeah.
But still, you have to look at it from an analytical perspective as well. Yeah, but it's quite cool with the states and so on. We try also this with control theory and everything, but it's quite hard when you're dealing with nonlinear dynamics. But
I just I think I would try out a hidden Markov model for the random walk.
Okay, cool. Thanks. Any more comments or questions?
Yeah, I have a question. Thank you for the talk. So if you do so many random walks, we know that random walks very much look like diffusion, and other groups have done diffusion. So what are the benefit or the limitation of doing one or the other, or the way you see it?
Yeah, good question. So it's similar to a diffusion model. It's just for us was more visually, we could better explain to the neurologists how this is progressing with this simple picture of the random walker, but mathematically it's the same. So it's just
so if you just integrate directly the diffusion equation over a long time, you'll get the same result that you have for your random walks.
Probably closely. So I haven't done that, but when we would have submitted to a journal with more network scientists, I'm sure we would have gotten that comment as well. So thanks for that. We definitely have to benchmark with other models which are similar. But the random walker here, the only advantage or difference I can think of is that it really stops after 5 steps, whereas the diffusion model is often hard to stop. So it's often going through just the whole network. And we wanted to really have it more local because this is what we see on the MRI scans. So
Okay. So in terms of you say you weigh your network. What do you weigh it with? What's the weight of your connection?
The weight of the connection is here the number of streamlines. So it's just a count of how many of the streamlines go really all the way from this one region to the other region. And
you don't scale it by the length at all?
No. But often there is a strong correlation between the length and number of streamlines. So the longer a connection, the smaller the number of streamlines. So we have a correlation there. But it's just because of the measurement. So it doesn't often is the case in the brain, but the measurement technique, it's the case that longer streamlines are harder to track, and you will end up with tracking less streamlines that go all the way from one region to the other. But distance is definitely something we have to look into.
And just one last question. You do have if I remember from the early paper by your group, and I think you said that you also have an autocatalytic process, right? When you see it at the location when you visit them with a certain probability.
Yeah.
So it's not just a simple random walk. It's a random walk, because a random walk would just give you diffusion, whereas if you have an autocatalytic process, you have a nonlinear dynamics on top of it.
So autocatalytic process, I don't know the word, but we start in only in very specific regions only. So we don't start all over. But
what I mean by that is that if you visit a site, then you can reseed that site. You can multiply the number of walkers. You don't
exactly.
So a simple diffusion process should be completely driven by the source where you start. But the autocatalytic process, you start with one. It's self-templated by a prion-like mechanism, like you discussed, which is an autocatalytic process, and you create more, right? So you do have a nonlinear process of growth.
We do, indeed. Yeah, thanks for explaining the word. Sorry, I didn't know that.
No, that's okay. It's different, as you say, it's just a matter of jargon.
It's vocabulary.
So really. Another more pointed question is, so there are different probability associated with either transport due to diffusion and this amplification of the seeds. And there's been a lot of discussion, especially for Alzheimer and other, whether or not the process is growth dominated or diffusion dominated. So if it's essentially early on, you would first rapidly explore the entire brain and then slowly increase, or you would just increase in one zone and then invade the next one by transport. Do you have any thought on that?
I think the second, so that you mentioned, is more true for ALS, because when we look at this post-mortem data, we see that not the whole brain is invaded by PTDP43, but they really make tests and they find that it's very localized in this disease compared to Alzheimer's.
Okay, so a very, very last question since
sure.
So there is something in ALS that shows, and that's been known somewhat for many years, is that there is very little damage in the back of the occipital lobe, the superior part of the occipital lobe. Is that something that your model can explain? Is that something you recover, or by diffusion you essentially, the connectome doesn't stop there? So is there a process by which it's you see that?
Yeah, I would have to look up for that specific region what we or that lobe what we had there. And if we just look at it, we can see that it's kind of spared, as are other regions. So we can see, let me not go back all the way, but we can see that it is that some regions are spared. And the hypothesis for that would be, if it's now the case for us as well, that it's really just on the connectome, too far away from the random walker. Since we find that 5 steps is optimal, it could be that this is either going over a longer way, so not well connected to the initial regions where we start, so not as well connected to the source. Or maybe there's some protective mechanism that's just some strong links leading away from this region very fast. That could also be. But
it's not the case for structural, because if you run with any other disease, you'll always invade that region, so you wouldn't stop. But your explanation seems very, very interesting, that it's not actual diffusion, it's kind of a limited diffusion. Because the diffusion process, you have non-zero concentration at every point at any positive time.
Yeah, and we
okay.
can have zero concentration. Not if we do it 1,000 times.
You have much more like a compact front invading, actually, right? In terms of continuous dynamics. Okay, that's very interesting. Thank you. Thank you so much.
You're welcome.
Can I ask a question again? Or is someone else? I don't know if someone else wants to ask a question. But if not, you know, you mentioned these when you form the network with the p-values to get the strongest connected components. Why is that done? Is it done for purely computational efficiency because to make the network more manageable, or is it done for other reasons?
It's really done because we would have to use multiple comparison correction, like Bonferroni or something, family-wise error, because we are testing so many different links. So this is really just done to avoid testing so many different links and really just narrowing it down, let's say, to some specific ones.
Right, okay. Okay, so to kind of pick out where the control and the normal patients differ, the diseased patients differ, essentially.
Yeah, it's really just finding the sweet spot or something where you really have the cluster of most connected, of differences. Yeah.
One final question, if it's okay. I know it's past 3 o'clock, but beyond, have you tried other models beyond well, not necessarily random walk, but kind of other spreading models? Because I'm working on something and I've been looking to kind of go into the neuroscience space, but I haven't had a chance to yet. But I was wondering if you have looked beyond, this random walk model that you've got so far.
So if for this study, we haven't really looked beyond because the model was already fitting so well. But for other studies I've looked at, where I've looked at brain dynamics and just trying to replicate the functional networks, I've looked at epidemic models as well, for example, which seem to also describe the functioning of the brain in a very simplified scale, but they describe it sufficiently as well. So you can think of so many different models, and I think that you would have to benchmark them all before being sure which model it is. Yeah.
Okay. No, thank you. I'll probably shoot you an email. One final question.
Sure.
Is the data set you worked on, is it available or is it not? I don't know.
Yeah, it's still one of the tasks to make it publicly available. So it's currently not open source or something, because it's such a huge value of having so many patients. It just gives you an advantage in the field of ALS imaging. But it's definitely open for collaboration. I can put you in touch with the people to make you use just the connectomes, because they are ready already, So if you want to just start from the network, this is definitely something we can talk about. Okay,
cool. I'll shoot you an email then, hopefully. Thank you.
Yeah, because then it's all anonymized, that's something we can talk about. If you want patient data, it's always complicated.
Yeah.
If you want the MRI scans, you can reconstruct the face, so that's a problem, right? Yeah.
Okay, great. Thanks everyone for the questions, and of course for Jill for answering them so clearly. I think we're running just out of time, so I guess it's time to wrap up. And thanks again, Jill. It was super
Thanks everybody for the great questions. That seemed like it really got across something, so I'm really happy.